min sururi anfusina wa min sayyiati amalina. May yahdihillahu fala mudhillalah wa may yudlilhu fala hadiyalah. Asyhadu an la ilaha illallah wahdahu la syarikalah wa asyhadu anna Muhammadan abduhu wa rasuluh, amma ba'du. The Honorable the Vice Rector for Academic and Partnership Affairs UMP, Bapak Saifur Rahman PhD. The invited deans, especially Dean of Faculty of Teacher Training and Education, Dean of Faculty Pharmacy, and also Dean of Faculty of Agriculture, who are the organizers of this International Seminar 2024, and also our IT and Public Relations team. My sincere gratitude to our special guest from Hainan University, China. We have Professor Jie Luo and Dr. Xi Hua, whom I know that this is their very first visit to UMP, and hopefully it is not going to be the last. Professor Jie Luo will talk about present research progress on the Chinese coconut industry and multi-omic studies. So welcome to UMP, Professor Xi and Professor Xi Hua. We also have Professor Sisunandar from Biologi Education Study Program, who also going to be the speaker in this international seminar. Thank you for being with us, Bapak. We are so happy that we have so many students here. Of course, students from Biologi Education Program Study, Faculty of Pharmacy, and Faculty of Agriculture and Aquaculture. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, please welcome to this International Seminar 2024, Research Progress on Coconut and Multi-omic Studies in China and Indonesia. First of all, let's thank to Allah Subhanahu wa Ta'ala for His blessing, so we can gather here together. Second, praise and salutation to our Prophet Muhammad Sallallahu Alaihi Wasallam. So before we start the agenda, let's recite Basmalah together. Bismillahirrahmanirrahim. Well, ladies and gentlemen, to start the agenda today, let's hear Al-Quran recitation by Seviana, our talented student from Biologi Education. Dear Seviana, the stage is yours.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم قد أفلح المؤمنون الذين هم في صلاتهم خاشعون والذين هم عن اللغو معرضون والذين هم للزكاة فاعلون والذين هم لفروجهم حافظون إلا على أزواجهم أو ما ملكت أيمانهم فإنهم غير ملومين فمن ابتغى وراء ذلك فأولئك هم العادون والذين هم لأماناتهم وعهدهم راعون والذين هم على صلاتهم يحافظون أولئك هم الوارثون الذين يرثون الفردوس هم فيها خالدون صدق الله العظيم. Assalamualaikum warahmatullahi wabarakatuh.
Risa, please come to the stage.
ia beriman membuahkan sejadah berbudi, beramal membina masyarakat adil dan makmur di negeri ilahi. Maju kembangkan selera ilmu, harumkan biru pertiwi, suburkan ukhuwah Islamiyah, sejahtera umat sabumi. Ya Allah, sedih meneguhkan ikrar dan janji prasetia kami. Kami memohon taufikmu ya Rabbi. Kami memohon taufikmu ya Rabbi.
All audiences, please be seated. Ladies and gentlemen, this is the moment that we've been waiting for. The welcoming speech and official opening of International Seminar 2024 Research Progress on Coconut and Multi-Omics Studies in China and Indonesia by Bapak Saifur Rahman PhD as the Vice Rector of Academic and Partnership Affairs of UMP. Everyone, please give us big applause to Bapak Saifur Rahman PhD.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. The Honorable Professor Jie Luo from Hainan University, welcome to Universitas Muhammadiyah Purwokerto, Prof. The Honorable Professor Sisunandar, one of our best professor in UMP, in which the research is about the Coconut Center, and then also coming now, the Dean of Faculty of Teacher Training and Education, Pak Dr. Eli Hasan Sadeli, and then also the Dean of the Farmasi Faculty, Bu Dr. Binar, and then also some of the heads of the departments in Universitas Muhammadiyah Purwokerto, from Argotek, and then also from the Biology as the host, Bu Yuli, Bu Dr. Yuli, Pak Arif from Argotex, yeah, and then also and the honorables, the committee, the head of the committee as well as the head of the international office, Pak Dr. Chandra Nur Alim, with the staff from the international office, and all of the audiences of this international seminar about the research progress on coconut and multi-omics studies in China and Indonesia. Ladies and gentlemen, let's thanks to Allah Subhanahu wa Ta'ala because of His blessing. We can come today in the international seminar, and salawat and salam be upon to our best Prophet Muhammad Sallallahu Alaihi Wasallam. Prof. Jie Luo, so now in our university we are developing the science technology, especially related to the biology and also other what's that, the planting probably, yeah, and then the pioneer is from Professor Sisunandar, in which that the research is about the kelapa kopior using the tissue cultures. And then for the shortcoming, I think that we also want to develop some of the innovations in our technology from Agotrek, yeah, Pak Arif, yeah. If I'm not mistaken, it also will have the durian without the seed. Yeah, as the, why is that, our excellence, yeah, durian, you know, Prof, durian, so we have so many durians, and then using a tissue culture, also one of our lecturer want to develop in. And then also in the pharmacy, we have also developed some of the herbal medicines, and then we have so many ways that innovations on the herbal medicine. I'm sure that the China is the place, right, of the herbal medicines. And beside this international conference, also, Prof, we do hope that we can, do more cooperations or collaborations between Universitas Muhammadiyah Purwokerto and then Hainan University. So after this, after the conference, the Dean will talk to you about the cooperations, and hopefully that we can also have the lecturers exchange and then also the students exchange. Ini mahasiswa mau pergi ke Cina semua? Betul. I'm sorry, I'm using Indonesia. I asked them, do you want to go to the China? And then they answer yes, Prof. Yeah. Iya, nanti sebentar lagi kita akan ada, moga-moga ya, lecture ya Pak Chandro ya, lecturer and the students exchange bagi mereka yang terpilih. So we will do a lecturer and the students exchange ya. So we have the wise word here, Prof, in our, as a Muslim, Prof. So our, yeah. If you want to enlighten your science, please go to the China. Ya, pergilah ke negeri Cina untuk menuntut ilmu ya. Jadi, so I think that I do agree with this wise word, because when I told to you when I was in Guangzhou also, mahasiswa ya. So almost in a university we cannot see the motorcycle or bikes like this or the cycle there. So everywhere in the university they walk, yeah, and they talk about science, they talk about knowledge and everything. And then I think that this is a good practice that we can have from the China. And Prof. Jie Luo and then also Prof. Sisunandar, hopefully by this international conference, our lecturer, our students can have more knowledge, especially dealing with the research on the coconut centers and then also the progress of the coconut center. And then by saying Basmalah together, Bismillahirrahmanirrahim. So I officially open this international seminar. Thank you so much and have a nice seminar. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Bapak. Ladies and gentlemen, before we start for the next agenda, let me invite the invited guests to come forward to the stage to have a photo session together. Bapak bisa welcome to the stage. We're gonna take a picture first together, Prof. Thank you, Bapak. You may come back to the seat. For Bapak, and then for the students, we will have photo session together. We will You may come to the front. Mahasiswa boleh ke depan buat foto sama profesornya. Oh, stay aja di sini. Thank you. Let's have a seat.
Nanti kamu nggak usah manggil moderator, langsung memperkenalkan moderator aja ya.
So in this seminar, International Seminar 2024 will be moderated by Ibu Ika Nuriza. Before that, I will read her CV. So Ibu Ika Nuriza is a lecturer and a researcher of Faculty of Pharmacy UMP. Ibu Ika Nuriza gets her master's degree at Wageningen University, Netherlands, in medicine and science. Then she continued her study at veterinary medicine and science at University of Nottingham, United Kingdom. She writes a number of scientific journals in collaboration with other researchers in medicine and science scopes. Ibu Ika is awardee of LPDP scholarship, and she also achieved some international rewards, such as Best Poster Presenter, Rothamsted Annual PhD Symposium, Oral Presentation Prize winner, Avian Research Symposium from the University of Edinburgh, and also honorary mention, Flash Talk Award, Cara One Health Poultry Conference from the Pilbright Institutes. I'll give to the moderator. To Ibu Ika, the stage is yours.
Thank you very much for the MC for very kind introduction. I'm not really that great. It may look on the paper, but I'm just an ordinary lecturer, unlike our esteemed professor here. So again, thank you very much for the organizing committee allowing me for chairing this session this morning. Before I begin the session this morning, of course, I would like to introduce our esteemed speaker this morning. So we have two distinguished professors. We have Professor Jie Luo, sorry. Professor Jie Luo, sorry. He's currently professor at Hainan University at the College of Tropical Crops. And his area of expertise comprising the understanding of the synthesis, regulation, and the function of a plant-derived natural product. Also focusing on the genetic basis of the metabolic diversity and its role in adaptation and also evolution, gene expression regulation, function diversification of enzyme, signal transduction of external and internal stimuli, and interaction between different pathways. It's a very complex study, I believe, but very beneficial for a plant science study, I believe. Also, that's our first speaker, Professor Jie Luo from the Hainan University. And Next for our second speaker, I'm sure everyone here has already known him, Professor Sisunandar, a very beloved lecturer at the University of Muhammadiyah Purwokerto. He's currently head and also founder of Coconut Research Center in Universitas Muhammadiyah Purwokerto. His research is focusing on copior coconut. He's also holders of several intellectual property rights. Professor Sisunandar has been published in many renowned national and also international journals. So that's a short introduction from our speakers this morning. So I think without further ado, I will give the floor to our first speaker, Professor Jie Luo from Hainan University. So you may stand in the podium for delivering your presentation, so time is yours.
Thank you. Good morning, ladies and gentlemen. So I'm very happy, pleased, honored to be here. And so last year, we were very pleased to have Professor Sisunandar in Hainan, in our university. And today, I'm very pleased to be here. And I would like to thank the introduction and also the invitation from Professor Sisunandar. So my talk today will be, I didn't know that we'll have this kind of a seminar, so maybe a little bit not related to the our theme, but I'll just talk a little bit about what we were doing and what we are doing at the moment. Because I'm actually the head of the Metabolic Biology Laboratory in Hainan University. I spent a few years in the United Kingdom at the Joint Illness Center. I'm not sure whether any of you have heard about that institution, or it's kind of the southeast part of UK, and it's an institute mainly focused on plant and also microbiology, kind of the science. So I did my postdoc there. So my name, I think it's a little bit difficult for you to pronounce, but you can call me Roger. Because the Chinese name is exactly pronunciation as the Roger. So my name is actually Luo Jie, Roger. That's the way that English people call us Roger. Easier. Yeah, I'm at Hainan University. So how can I move forward? Or all right, good. Yeah. So first a little bit about my team. So I have a team which work mainly on plant science or more specifically crop science, and we are all working on the metabolic regulation and also the metabolomics. So we have about over 10 say members or teachers in our team, and have over 100 different students in our team. It's a kind of a big team. So we have worked on this for over 20 years, and we have published a number of papers in Cell, in Nature Genetics, in a bunch of different journals. So we are mainly working on metabolites. So just a very brief introduction of what metabolites and what metabolomics. So we all know that plants produce a huge amount of different metabolites, mainly because they are C-cell species. They can't move, right, in response to different stresses, either abiotic stresses or biotic stresses. So they can't move, so they have to make a lot of different metabolites to cope with different stresses, right? There are how many of these plant metabolites? They're estimated to be 200,000 to 1 million. So that's a huge number. But they can be divided into two big categories. One is called the primary metabolites, right? So you are very familiar with those ones. Sugars, sorry, no? Yes, sugars, amino acids, fatty acids, they are all primary metabolites. They are existing in every single plant species, right? They're everywhere, they're universe, so they're called primary metabolites. And these metabolites are supposed to be necessary for the growth and development of all plant species, all right? In addition to that, we have even larger number of another category of metabolites, which is called secondary metabolites previously, but we now call it specialized metabolites because everybody wants to be special. They don't want to be secondary, all right? So we call it specialized metabolites. And the scientists and the people work on specialized metabolites. They're special scientists, that's me, right? So we want to be special. How many of them? They are supposed to be 200,000 or 1 million. So majority of metabolites are these specialized metabolites. So in terms of the primary metabolites, we are talking about thousands or tens of thousands. But for specialized metabolites, we are talking about a number of hundreds, thousands to million, right? So even though the big number, they can be further divided into 4 subgroups. One is called the phenylpropanoid. I'm not sure whether you're familiar with this, but you must be familiar with the flavonoid. So you drink tea maybe every day. Tea has a lot of these flavonoids, and these ones are also called polyphenols, right? Polyphenols. So this is the first one, and the second one is called the terpenoid, right? I'm not sure you're familiar with, but carotenoid, right? You can see this red color for tomato is the terpenoid, all right? We have this carotenoid and the steroid, and you can see this purple color, right? But what's the difference between these two? It's different color, of course, but it has a different group of metabolites. So this purple one is actually phenylpropanoid, the anthocyanins, but this one is carotenoid, the terpenoids. They are water insoluble, but this one, the purple one, is water soluble, right? And the third category is alkaloids. We have this indole you must be familiar with, and this is the fourth group, is the sulfur-containing compound. You have this glucosinolates from cauliflower. You must be familiar with that. They have kind of the very special smell, right? So you have this sulfur-containing compound, right? So, and I mentioned that there's so many numbers of different metabolites. So how can we deal with, how can we research on this great number, huge number? We use metabolomics, right? We use metabolomics to study all kinds of different plant metabolites. So what is metabolomics? Can be defined as an analysis of all metabolites in a biological sample, right? So in an organism or in a biological sample. So there are basically 2 methods that you can analyze different metabolites using metabolomics. One is called non-targeted metabolomics. So we look at, say, for example, we look at all the people in a hall in this room. We are not specifically targeted to anyone, but we just has a glance of all this. So it's kind of a non-targeted, but we also have targeted. So we may target a specific person, and we also can target specific metabolites as a strategy to use the metabolomics. So the advantage and the disadvantage of both this method. So for the targeted one, I'm not sure whether you can see that. So for the targeted metabolomics, what you can do that you can measure a specific metabolite or some metabolites at a time, but the number is very limited, from dozens to 100 normally. You can measure them very accurately and very sensitively. So that is to say, if I specifically target to a person, I can get very familiar with this person, right? So I can identify this person very accurately, right? So that's for the targeted, but the problem with that, you can only target a small number at a time, usually, right? So the number is limited, but you can examine the accuracy and also high sensitivity. And the untargeted one, you can measure a large number, say hundreds to thousands of metabolites at a time, but you can only measure the ones that are has very high abundancy. So you can't measure the metabolites at lower concentration. I'm not sure whether, am I make me understood? It's okay? Yeah. So these two have both advantages and disadvantages. So we then develop a method. We can measure as many as metabolites as the untargeted, but at a sensitivity equal to the targeted ones. So we call it the widely targeted metabolomics, right? So we can measure thousands, more than 1,000 different metabolites in about 15 minutes at the sensitivity and also the accuracy as the targeted one. So with that, we can do this. And we can also set up a method which is called the widely targeted volatomics, in which you can measure all the compound that has smelly compound, right? The volatiles and the non-volatiles. So in terms of the metabolomics, we can say that we can measure everything, either non-volatile or volatiles, right? So with this method, what we are doing that we can generate populations of different plant species, say rice, say maize, and say coconuts, right? We generate populations of that, and then we can sequence every single plant to get the biomarkers, right? To get the biomarkers for each individual lines, and we can also measure the metabolites and also these phenotypes, say the height, the size, the yield, right? All these are economical, agronomical traits, and then we can use the correlation analysis, either QTL analysis or association analysis. I'm not sure whether you're familiar with those analyses. And the reason we want to do both genomics and metabolomics, we can combine these two omics to find the locus or the locus or the loci that control specific traits using this association or linkage analysis, right? So maybe I can say in an easy way, a very specific trait, say for the plant height, use this population, we can get the locus, loci, and also the gene responsible for the different height of individual plants, right? And this is also true for yield, for anything you can see, or for anything that you can't see, but can see by the machine. Say we have a different content of sugar, anything can do that, right? With that, we have published a number of papers in different journals in the last about 15 years. So I just very briefly show some examples of that. So we generate a population of different rice around the world, and we can use this QTL analysis to find the locus, right? You can see here. Sorry. I can't. Yeah. You can see here, this means there's a gene here that is responsible for kind of this secondary metabolites or specialized metabolite, and we also have this, and we can also investigate the different interaction between different loci within the population. So this, if it's controlled by a single locus or two loci, and the interaction between these two loci. And this is also true when we generate the kind of the natural population. So we collect the different varieties, different accessions around the world of different rice, and can use this population to dissect the genetic basis of, say, yield, say, plant height, say, different metabolites, anything you can measure. We can then find the genes and also the locus or the markers that you can use for the crop breeding, right? So this is just the proof of concept of the strategy we can use. And also what we can do is that we can use this strategy into the crops that have a very big genome. So for maize, it has about more than 2 gigabytes of a genome. It's a complicated genome. We can show that this strategy is also applied to plant species with very large and complicated genomes, right? And we can also even further show that we can then use this strategy, modified strategy, to combine the rice and the maize to kind of dissect the diversity, not just within species, but also between species, right? So, I mean, so I don't have time to explain too much detail. And for this one, it's a very short story about the domestication of cultivated tomato. So you have, I'm not sure whether you have known about the wild tomato. It's a very small one with a weight of 1 to 2 grams. And after domestication, you have a kind of the medium-sized tomato, which is about 10 to 20 grams per fruit, right? But nowadays, we are eating the big tomato with about 100 to 200 grams per fruit, right? So this kind of called the domestication and also the improvement of tomato. So we know the genes already responsible for the increase of fruit size, right? Or the fruit size. But do we know what happens during tomato domestication? What kind of metabolites has changed? People don't know that, right? So we are very interested in during domestication and improvement, what kind of metabolites have been changed, and what is the gene responsible for this change, right? And then we use this genome-wide association study to find that this is actually the key locus, right, responsible for the decrease of what we call the glycoalkaloid, the beta compound in the wild tomato. The wild tomato used to be very big and it's harmful to our health, all right? It's toxic to people, to human, but can insect resistant. During domestication, you can see that most of this beta compound has decreased or even lost during domestication and the improvement. We then find the major locus that's responsible for this decrease, and also the genes that's responsible. Interestingly, there's only one nucleotide mutation between the wild tomato and the cultivated tomato. that is responsible for the loss of this beta compound. You can see here TGG changed to TGA, right? There's only G2A mutation, single nucleotide mutation, causes a premature stop codon. That means the loss function of this enzyme and also the loss of accumulation of this beta compound, right? I'm not sure, right? Everybody understand that? Right? Okay. That's a very simple story of domestication of tomato, right? And yeah, I'll just speed up a little bit. We also investigate different variation of this is caspin. It's a diterpenoid during rice domestication, right? Now a little bit about the coconut research. What we are doing now, so we are collecting coconut resources from Hainan, from China, and also we want to do it around the world to collect samples, right? And we would like to, yeah, we have generated, assembled the first, so far the best genome of coconut, both for the dwarf coconut and also for the tall coconut, right? So we collect the resources, we have the genome assembled, and we also investigate kind of the evolution of the different chromosomes of coconut, right? So we found that the coconut used to have kind of the, not coconut, the kind of the monocots have the karyotype chromosome of 10 rather than this 12 in coconut for today, right? And we also find the genes that are responsible for the difference between the tall coconut and the dwarf one. So have you heard about the Green Revolution? For rice, for maize, for cotton. So there's a Green Revolution gene, which is the SD1. It's responsible for the biosynthesis of GA, gibberellin, right? Do you know that? Should have heard of gibberellin? Yeah, this is a hormone that's responsible for the growth of plant. And if you lose this hormone or you have a decreased amount of hormone, you get the dwarf plants, right? And the dwarf rice is actually responsible for the Green Revolution. So this one is easy to fail off. So you get no yield for this one, right? But this dwarf one, they're resistant to wind. It can stay there, right? So you get higher yield. So this is called the Green Revolution. And this is, I think, maybe the only one crop agriculture scientist has won the Nobel Prize, right? Because we don't have agriculture for Nobel Prize. We don't have, but this one is the Peace Nobel Prize, right? This is the only one. And interesting, we found that coconut is the same gene that caused the dwarfing of the coconut, but this has happened 5 million years ago by nature. But this one, this Green Revolution, is happened about 50, 60 years ago by human selection. So you can see that the human being kind of set up a revolution, but it happened in nature million years ago, right? So this is the case. And we are, can't move it forward. Next. Yeah. And we can also use this strategy to dissect kind of the fiber formation during coconuts development. Next. Yeah. And we can also do GWAS for coconuts as well. similar to the things that we have done for other crops or other plant species, right? Please, yeah. And so we use this to dissect a number of agronomical traits, phenoms, and metabolic traits. Say we use that to dissect the enlarged tree base for coconuts, and also bundles, and also the fruit or the yield of coconuts. Please. Yeah, and we also use this kind of the micro CT to do CT for coconuts, right? Rather than do it for human body, we use it for coconuts. So we can use this one. We can measure different kind of the shape, the length, the size, the weight, and also the volume of the water within the coconuts, right? We can then use GWAS to dissect the genetic base of those traits. Next slide, please. Right, we also generate a database that we can use for kind of a multi-omics. All right? Okay, yeah. Just a few slides, okay, yeah. So that's the kind of the data we've obtained for the GWAS of different agronomical and economical traits for coconuts, right? So we kind of set up a platform that we can do systematic multi-omics for coconuts, as well as the other crops, right? So next slide, please. Right, yeah, this is kind of the database that we have generated. Yes, next. And Dr. Zhihua Mu also developed a tissue culture method. Yeah, with the help maybe from you. Yes, Sisunandar, we kind of developed a tissue culture method for coconuts using the blue milo, right? Next, please. Yeah, so I think this is the last one, potential direction for cooperation. So we would like to cooperate with you and the others to collect coconut germplasms around the world, and we also want to do the assembly of the coconut pan genome. We have 2 genomes already, but we want to have more than 10 or maybe 100 something genomes, right? We can do the pan genome assembly and also use this pan genome to dissect the genetic basis of other important traits. And we also want to do genetic work, the genetic diversity, and finally the tissue culture, and also maybe the transformation for the coconuts. And yeah, this is the last slide. So I just want to advertise our platform and our university. So we are recruiting PhD students, postdocs, junior scientists in China, in Hainan. So this actually the place, the island we are on is the second biggest island in China. It's very, very south of China, right? So it's kind of the nearest place to your country from China, right? So it's a tropical atmosphere, right? Tropical city, so very similar to here, but it's very beautiful as well. So with that, I would just thank you, and I'd like to answer questions. Thank you.
Discussion after the next speaker. Thank you very much, Professor Roger, for delivering excellent talk. I see that of the student participant here, I see that they're very eager to ask a question, but yes, we're going to save that question for later. Right, yeah, that's fine. You can just leave it there. Yeah, it's working. It's working. Yeah, thank you. Yeah. So we're going to have a discussion session after the next speaker. So please save your question for later. Before I begin and also move on to the next speaker, which is Professor Sisunandar, we're going to have a little ice breaking. Would you maaf, yes. Is it ready? So yeah, we're going to listen to a song called Rayuan Pulau Kelapa, and in English it's translated as Console of Coconut Island. Right, okay, yes. The solace of the Coconut Island, so it's described the, and also praising the beauty of this country, all of the flora, fauna, the plants, and describe this country as a coconut island. And also a little trivia about this song, for those living abroad. So this is sort of like a national anthem for them, to remind them of this country while they're living abroad. So, and also it's also talking about coconut, which is relevant with our topic this morning. So, so if it's ready, the song, we're going to listen to the song.
Yang ku puja sepanjang masa.
I hope everyone enjoyed the song. It's sort of the modern rendition of the song, so but it doesn't really remove the nostalgic feeling every time we listen to this song. So yeah, also hopefully you may be able to visit some of the landmark that is shown in the video. I don't know if you will have a time for that. Okay, so we're going to move on to our next speaker, which is Professor Sisunandar. So you may begin your presentation. You have approximately 45 minutes.
Yeah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Dean of Farmasi, Dean of Agriculture, Head of School, Biological School, all the staff and lecturer, student from Farmasi, from Biology, from Agriculture. First of all, I'd like to say thank you so much to Professor Jie and also to my best friend, Dr. Abraham Mu, that take more than 30 hours to be here with us. So I would like for you to give applause to them because they 30 hours, 30 jam to be here. It's a long journey from Hainan Island, from China, to be here. You have to fly to the north of China and then go to Jakarta. Yesterday, have to wait, delay for 3-4 hours, delay in Jakarta to go to Jogja. And just last night, they arrived in Purwokerto at 11. Mereka datang jam 11 malam tadi malam dan berangkat hari Minggu jam 11 siang. Jadi long journey to be here with you. Today I want to talk just a little bit about Kopior. What we have done and what we will do in the future. And it's not working. Okay, sorry. Yeah, this one is working. As we all know that coconut, not only Kopior, is very important for Indonesia. We just finished listening Rayuan Bulu Kelapa. It's the song of coconut in Indonesia. It's very important. We know that all of the part of the coconut we can use it for many things. With the traditional one, we use for building a house and for the roof and everything. And so this is charcoal, and for the furniture, and also the one used for oil, coconut oil. The virgin coconut oil is very famous. Even just drinking the fresh water, using for special day for kupat we call here, for when Idul Fitri finish and then we make food using coconut. And now the product from coconut is varying, starting from coconut milk, and coconut water is very popular. If we know that most of, but this is not our culture. Most of Chinese people now drinking coconut milk, some of them, but not with us. Mereka minum santan tapi kita tidak. Because we have a lot of important ingredient on santan, on coconut milk. We still not common here drinking santan, drinking coconut milk, but not with them. It's very popular in China. This is another product that this area, the Banyumas, is the most important production of coconut sugar in Indonesia. And we already sell to overseas, especially to European country and American. So coconut sugar is very important here. We also have many things produced from coconut. Indonesia is also a blessing country. Almost 1/4 of coconut germplasm is in Indonesia. 1/4. Lebih dari 100, more than 100 of cultivar in the world is in Indonesia. While around the world, we have around 400 cultivar. So Indonesia is really a blessing country. And we still have more than 400 cultivar has not been determined yet. It's need our hand to determine coconut, new cultivar in Indonesia. But that's the problem. Most of the coconut farmer living in poverty, because 96% of coconut plantation is belong to smallholder farmer, and the price of coconut is very low here. The price is just about 1,500 rupiah to 3,000 per nut. It's about 10 cents US dollar. And because the price is very low, then the farmer income is also very low as well. So, how to overcome? One of the possible approach that we can use to overcome the problem on lower income coconut farmer is by planting special coconut. We have copier coconut in Indonesia that didn't have in other country. If we are not quite familiar, because probably some of you still not quite often open coconut. On the other side is the normal one, that the kernel, the endosperm, the solid endosperm, dagingnya is hard, and we usually use for santan, we usually use for producing coconut oil, for cooking, and the cavity is full of water here. If we open coconut, usually we got the coconut water. Actually, coconut water is liquid endosperm. It was solid and then changed to become liquid. It was liquid and then changed to become solid while they are developed. For the very first, they are all is liquid, but during the development process, then from liquid change to become solid. So it is why inside the cavity you can find two kind of endosperm, solid and liquid, but not the case for the copier. Most of copier starting from liquid, and then during the development process, most of the liquids change to become solid. So we rarely found, it's not really common, we found the coconut water from copier. So most of them is like that, all in the cavity full of the solid endosperm, not the liquid one. And for farmer, the most important one, the price for copier here is quite expensive, 30,000 to 60,000 rupiah per nut. So it's been around how much? 2 to 5 US dollar, compared to 10 cents US$ per nut. So it's more than 10 times higher than the normal. So it is why if we use this technique or farmer cultivate copier coconut, then big hope there. Later on, farmer can have more income rather than the normal. The problem with copier coconut, if for the normal we can use the fruit and producing seedling and then planting to the field and then so on, but not with the copier. It's not possible for planting using their fruit because when we harvest the nut, after 2 or 3 weeks, then it becomes stink inside. It's not possible to support the embryo to growing. So it is why we need technology here. We developed embryo culture starting from 2008, and now we are successfully producing seedling and putting in the field, and now we have coconut copier gene bank in our university. Actually, all students can access there and can study using this special material. But I do hope that some of you join to do research using copier. This is the method how we work with to produce a seedling. We collect the embryo, put in the tissue culture, growing them, and get the seedling after 16 to 19 months. But so for undergraduate students, when they want to do their research for their thesis, that is not quite good time. I mean, you have to start a little bit earlier because you have only 1 semester or 2 semesters for your research, but this one need 2 years to produce the seedling. And we already planting in the field. Now we have more than 14, it's about 14 varieties. We have yellow, we have green, we have orange varieties, all copier, all coming from tissue culture, and all already in the university coconut gene bank, just in the front of asrama mahasiswa. So we have green dwarf, 4 kind of green dwarf we have. One is a really green dwarf, and then green with the brown one, and then we have special we call entok. It's coming from Kebumen, but there are 2 kind of copier entok coming from this area. One is coming from Kebumen, the other is coming from Banyumas. The shape of the nut is different. And we have a brown dwarf, 2 kind of brown dwarf there. And also the tall is very different one and other. We have 4 kind of copier tall. Oh, sorry, 5 copier tall. So the second one, it just need 4 years for producing seedling. The second one, but for the other is usually need 4 to 5 years for producing seedling. But the third one is the second one is very quick producing seedling. But this is tall. So we have 14 varieties already. We work a lot on agriculture as well, because copier is very sensitive to fertilizer. So for the one who want to study fertilizer, then you can use this one. You can use copier as a model, because when we have copier with less fertilizer, then the shape of the nut change from round to become, sorry, from round to become like this. And there is nothing inside. There is no sound, there is no endosperm, just as you can see here, just like that. It is because the fertilizer and water. When we put water and we put fertilizer, then back to become a normal nut. It was like that. And when we put water, we put fertilizer, and then change to become like that. Not the same nut, but the next nut produced change to become normal. So this is a really model for agriculture person who want to study fertilizer. The kind of copier is also varying. Some of them is just like normal coconut, but when we take the salt in the sperm, it's very soft, just copier, full inside with water, but most of them is like that. Inside the cavity is just full of solid endosperm with less of water. So we found many kind of copier surrounding Indonesia, and we already put in the coconut gene bank in this university. Oh, sorry. And we try to fulfill what coconut farmer want. Usually they want to have the good one. Good one is big fruit, but inside the cavity is full of solid endosperm. There is no water there. So most coconut farmers want to have that one. So we do crossing between tall and dwarf. For the normal, it's very easy, and this method has been applied in many coconut countries for normal. But we work with copier. Starting from 2020, we cross between tall and dwarf copier, and little bit complicated with copier because we can't put the nut directly to produce the seedling, but we still need embryo culture. So after we cross between tall and dwarf copier, and then we go embryo culture, going to the lab, waiting for another 2 years for produce seedling. But we successfully produce seedling already. We do a lot of analysis using morphological analysis. We work with molecular analysis as well. And we found that most of our hybrid copier is really a hybrid based on the morphological and molecular analysis. We found 1, 2, 3, 4, 5 seedling is not really hybrid. We do hand pollination. We take the pollen from male and then put in the female, but from almost 50 seedling we got around 5 seedling. It's questionable. It is really hybrid or not? So we do molecular analysis to make sure whether they are really hybrid or not. We found 3 of them is really, this is not hybrid. 3 of them. But the rest, they are all hybrid. So it means the hand pollination we do for producing hybrid copier can be applicable by farmer. It's easy. So most of farmer, if they want to produce hybrid, then they can do this method. It's quite quite easy, and even there is no education there. As long as we train them to do cross-pollination, that they will do that. It's very easy. But for the result, we have to wait until the next 3 years, because we planting in the field already last year. So we wait for another 3 years for checking whether they already have a better quality or just similar with other. Better quality means the nut is full with the solid endosperm, because we sell the solid endosperm as well. So most farmers want to have the more solid endosperm rather than the liquid one. We do analysis for fatty acid content. We found between coconut, common coconut, the normal coconut, and compare with the copier endosperm, and then compare with the copier liquid. We found quite interesting that normal coconut in the water usually only about 2.8% lauric acid. We all know that lauric acid is middle chain fatty acid that has the good impact for our health, especially they have antivirus, antimicrobial activities. So for the normal coconut, the water is only have around 2.8%, while for copier they have 34% of. So more than 10 times higher lauric acid compared to the normal for the coconut water. And for the endosperm is also the same, similar between the coconut water and the endosperm, the solid. The content of lauric acid is very similar. This condition, if we compare with the virgin coconut oil, even a little bit lower, but remain high. I mean, the VCO, the best VCO we analyzes have 49% of lauric acid, while copier solid in the sperm have about 30%. So we can recommend to you that if you want to consume virgin coconut oil, it's better to consume copier solid or copier water, because the content of lauric acid is quite similar. When you consume virgin coconut oil, the taste is not nice. I mean, because it is oil. You can drink coconut oil, but you can't have any taste on your mouth. But if we have if we have copier endosperm, the taste is very different. So later on, we produce couple things. We sell the nut, we sell the seedling, we sell the frozen copier, and also we sell the water of copier already. But most of the frozen copier, the solid endosperm, put in the freezer. Why we put in the frozen? Because when we harvesting on the whole nut, it's need only, we can only put about 2 to 3 weeks, and then we can consume it again. While when we collect the solid endosperm put in the freezer, it can last for more than 1 year. So 8 to 1 years, 8 months to 1 year, we can put in the freezer, and when we need it, and we can consume it. Most of them goes to for drink, for ice cream, for cake. Most of them. But we are waiting for your hand from the pharmacy for working with cosmetic, because it's already soft. The content is very good. So if we start producing them for cosmetic, for face masks, and so on, it's very good. And also, this year we are starting working just to use copier for baby food. So I mean, because copier have a very good ingredient, so when we can change them to become supplement for baby after breastfeeding, and then it will be very good. Especially we have a problem with standing. Some baby need more energy for the lower people, lower income people that need a lot of energy, a lot of ingredient that can be supported by solid endosperm of copier. And because the water of copier also has the high ingredient of lauric acid, and now we only collect the water, put in the freezer, it can last for about 3 months. So when we sell it, it's quite difficult because we have to sell it during frozen. So we are still looking on how to use the coconut water and put on the pack like hydro cocoa or FIFA cocoa and so on, so we can sell it easily. So this is what we did. Hopefully, the more hand working with copier, I do hope that my friend Professor Loji and also Dr. Mu will help us to develop this activity, to do research together. We are very glad that if we can do research collaboration together. Also, friends from Farmasi, from agriculture, to work with developing product for cosmetic, for food industry. Thank you so much.
Thank you very much, Professor Sisunandar, for the excellent talk. And I would like to invite back Professor Roger. We're now arrived at the discussion session, so I would like to give an opportunity for the participants here, for the students and also colleagues, to ask a question for our two speakers here. Are there any questions for the speakers? Yes.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. My name Arif Prasadi Santosa. I'm Pras. I'm from Agriculture Faculty, Agrotechnology Department. Thank you very much for the opportunity. First, I'll try with English, but if maybe you all don't get the question, I will repeat with Indonesian. Okay. So the moderator can translate for me. But actually, there is so many questions about omic because for me it's very new information. Maybe it's a very long knowledge about omic, but for me myself it's very new about omic. Then when you say when you say before, every plant, every commodity has specific metabolite. So is metabolomic, when we use metabolomic is when gimana ya, pada saat kita menggunakan metabolomic, apakah nantinya semua senyawa itu akan teridentifikasi? If are they all the metabolite of the plant is categorized or maybe recognized already all, or maybe or it's specific, as you said before, there is phenylpropanoid and then steroid, glycosanoid. Is it directly sekaligus keluar one in a moment, or maybe we can separate, phenylpropanoid first, then steroid, like that. Then for the there is a targeted and untargeted. When targeted is there is a standard or no. When we untargeted, when I say the untargeted is so many metabolite. Then how we know the targeted? Maybe I think like this. Pure coconut and regular coconut must have specific metabolite that make it different. How we know the different, how the gimana ya, mana metabolit yang paling berperan gitu. The metabolit that very distinguished to make maybe for the beneficial or for healthy and something like that. I think that for Mr. Roger, I mean, then for Mr. Prof. Sisunandar, there is coconut, dark coconut. Dark coconut already has found it, yeah. Maybe right now the production of coconut for the focus, but in Banyumas, not just the fruit of the coconut, but also the coconut sap. The coconut sap is a dwarf coconut tree produce coconut sap, menghasilkan nira tidak, karena saya pernah melakukan pelatihan gitu ya. Rata-rata para petani itu, petani gula kelapa atau pengrajin gula kelapa, memang yang dicari adalah kelapa atau pohon kelapa yang rendah, sehingga tidak perlu deres. Deresnya itu tidak terlalu tinggi. Nah, pada saat ini merupakan suatu hal yang sudah ditemukan, apakah memang produksinya dari si kelapa yang dwarf tadi ya, yang apa namanya, yang kerdil, apakah menghasilkan juga coconut sap, also produce coconut sap that also as good as regular coconut tree. I hope to you, Mr. Roger, do you get my question? Okay. I think that's my question. Thank you.
Right. Thank you for your question. So you have two questions, right? For the omics, metabolomics, right? So the first one is, as long as I understand, how many metabolites can we measure, right? In a sample, something like that, or?
Gini, mungkin justru makan nanti ya Bu. Jadi,
so,
maksudnya pada saat kita menggunakan metabolomik, kita akan mendapatkan banyak sekali, langsung mendapatkan banyak metabolitnya, merecognize itu gitu kan. Right. We recognize so many metabolite. How we know which one the metabolite is very distinguished, or maybe how we know that metabolite is maybe beneficial.
All right, Thank you.
I think like
so as far as we can do now, so there's so many. I mentioned that hundreds of thousands or even 1 million for all metabolites. But for single species, what we estimated that they have 1,000 to a few, maybe 10,000 different metabolites within a sample, right? A specific biological sample have maybe 1,000 to 10,000 or 20,000, the maximum of metabolites. So for the metabolomics we have now, we can measure several thousand at a time. So we can't measure all the metabolites within a single analysis, but we can measure them using different methods to kind of measure most of the metabolites within a sample, right? So the method we are developing, what we have developed, we have measured the number is 1 or 2,000 at a time. So we don't need to separate each metabolite. We can measure them simultaneously at one time. So include the ones maybe you are interested, some of the health promoting metabolites, right? Maybe you are also interested in some of the hormones. All these things, we can measure them all together simultaneously within about 10 to 15 minutes. So that kind of, say, if you are interested in yield, in plant height, you can only measure 1 or 2 or several traits at a time, right? But we can measure about 1,000 different traits at a single time, right? So that's kind of very efficient. And our method is we have high accuracy and high sensitivity. For example, for most of the plant hormones, the concentration is too low to be measured by other methods. We can measure most of the hormones as well, in addition to the health promoting or the other metabolites, right? So that's made it possible to measure as many as you can and as high throughput as you can, right? It's kind of a revolution of the analysis. And then we can combine those with genetics, with other omics, and with also the molecular biology to combine all these different methods or strategies to do biological research, right? I'm not sure whether I have answered your question.
Then the next question is, for the targeted, is there is a standard to make from the so many metabolite, so we can get the specific metabolite. So is need a standard or no?
Yeah, it depends on your purpose. If you are only interested in a small number of metabolites, you can use this targeted one, right? If you are interested in a number of them, say if you want to know the relationship between plant hormones and maybe flavonoids, you have to measure them all together, right? And with that, you might need to the untargeted. But the problem with the untargeted, you can't measure hormones using untargeted, right? Because its concentration is too low. With our method, we can measure as many metabolites using the targeted ones. So we can measure plant hormones, anything you are interested in. In theory, in principle, we can measure them all together, right? So that's the difference between the targeted, untargeted, and also the widely targeted, right? Thank you.
Yeah, thank you very much, sir.
Okay, thank you, Pak Pras. The question is, it's very common when we are going to coconut farmer that they are producing sap. They're producing coconut sugar. They keep asking to have the short statue of coconut, so they don't need to climbing up the tree twice a day. Because when we want to produce coconut sugar, farmers should climb coconut twice a day, in the morning and then another one is in the afternoon. In the morning to collect the sap collected during the night, and in the afternoon is during the day. And another reason is because the, probably you're familiar with browning, because the sap will coming out because we cut the inflorescence, the flower, and it's naturally happen in the plant when we cut the tissue, and it has a response to produce phenol and then start browning, and because the browning and then stop the sap coming out. So it is why farmer have to climbing up to cut it again, and twice a day. So it is why they are asking to have a low statue of coconut, the dwarf coconut. But we have to say to farmer that it's not possible to have coconut that skip short. They will growing up. Every year for the tall coconut, it will increase their height about 1 meter, while for the dwarf will increase about 60 centimeter every year. So it's not possible to have coconut that they are short all the years, all under 10 or 20 years, not possible. They will growing up, even for the dwarf. So the key for farmer should be connected with the condition of the environment. I mean, when farmer has the fertile soil, lowland, and it's better to planting dwarf coconut. But if farmer has soil in highland, the plantation is in highland, the soil is not quite fertile, it's better to use tall coconut. And tall coconut, even they grow quicker, but they produce sap more than the dwarf. So it is really a choice. If we want to have more sap and then we produce more coconut sugar, we have to plant a tall variety. But it's grow quicker than the dwarf one. So the best one is just crossing them, crossing between tall and dwarf. So we got hybrid that they grow a little bit slower compared to the tall, but the all condition is tall. So I mean the production is high, but they are grow slowly, don't need higher fertile soil compared to the dwarf, and they produce more because the inflorescence is longer. So the farmer can cut a little bit longer. I mean, on the same condition, on the same time, in every single plant, farmer can have 3 to 4 inflorescence on the same time to be cut to produce coconut sap. So when we have tall variety, then the more flower compared to the dwarf, because dwarf has the flower of dwarf is quite short. So when they have to cut it, it will take a little bit shorter compared for the tall, because tall is long, for the inflorescence is long. So when they cutting, it will take a longer time. They produce more sap rather than the dwarf one. So which one is good? It's really depend on the situation where farmer has. If they are coming from lowland, yes, they can have dwarf. But when they have highland more than 500 above sea level, it's better to planting tall rather than dwarf. So that's my question.
Is it possible to make like a bonsai for the when the coconut tree is dwarf? Then I don't know how to make a bonsai. Maybe we cut every time, so make it no longer grow.
It's not possible because coconut is a rosette. It's Seterusnya, I mean, they keep growing. So it is why they produce fruit, they produce flower. Otherwise, if we cut, they just stop. They only have single meristem on the top. It's not producing branch on the like other trees because it is monocot.
Okay.
So that's why. Yes, that's coconut.
I get it.
That's coconut. Not possible to cut them and then waiting for another branch coming out. When we cut, we lose the meristem on the top, and then the tree is finished. Okay. Thank you, Pak.
And then one question for a student. Question.
Okay, thank you. So my question will be first for Professor Sisunandar, and then it's going to be related to Professor Roja. So, Prof. Sisunandar, so is there any study, or well, I'm not sure whether you are already studied the metabolism of all of your copior variants, if because it's very interesting to see the difference of in terms of the metabolite or the genomic and etc. Because it's going, if it's not done yet, then it's going to be great collaboration for you with Professor Roger, right? As we already know that one of pharmacy student also trying to use the oil of your coconut to, so basically we already published a paper about making the lotion from the oil that produced by Prof. Sisunandar, and then we also analyzed the content of the alpha tocopherol inside the copier, and we compare it as well with the normal one, and we see much different that the alpha tocopherol in the copier coconut oil is much higher than the normal one. So based on that, we developed those kind of cosmetic preparation lotion, and the patent one that we are still waiting for the granted patent is the mask. Yeah, so those kind of example that I'm sure that the metabolite probably my primary and as well as the specialized metabolite is going to be different in all of the probably the variants of the copier of Professor Sisunandar seedling. That's my question for Professor Sisunandar, and then Professor Luo. So, so if we want to perform the metabolomic studies of targeted metabolite, then can we do like special preparation just only to like sorting out the one that we are not targeted, or what kind of? So my question is, what kind of, is a bit technical, what kind of preparation that we have to do to perform the metabolic study if we want to perform the targeted one? Yeah. Genetically modified, perhaps to see the more expression of certain metabolite, is that the one that also approach that we can use for metabolic study? Okay, that's the question. Thank you very much.
Okay. Thank you, Professor Luo. Thank you, Bubinar. Is there any study about metabolite on copier? The one that I found paper is about I think about 1990 something. They study about the content of sugar, content of protein, but when I read in detail, they didn't use good sample from copier. They just go to the market, find copier, and then analyze them. Why I know that? They said that copier is they use for sample is 7 months old, while 7 months old is there's nothing there. So probably they're just guessing. 7 months old is remain water inside the nut, still not develop any solid endosperm inside. So this literature is not really good example for us. We need more study that working very close for the metabolite comparison between copier and normal, and even to compare the development process from 3 months, 6 months, 8 months, what changed during their development, because the difference between normal and copier, for copier they can't stop producing solid from liquid, while for normal, about 9 to 10 months after they producing solid, then they stop. No, we never found all liquid change to become solid or normal. But we can find easily it's happened in copier. So there are many questions that we have to answer about what happened in this copier. If each variety has a different taste, different amount of solid, different amount of water, even for every single variety is different as well. So we really need a lot of hand to work with this special Indonesian mutant of coconut. This is, we up to now we can found only in Indonesia. We need, of course, Professor Lojo will help. Dr. Abraham also will help us. I'm very sure. It is why they are here to do this research together. So hopefully in the future we can produce a lot of information for farmer, for community, for other scientists that we have a very special condition here. Not much, Bu. The last paper produced is 2024 by this year, this month, but I haven't read yet. Copier is published by lecturer from UNS that she studied PhD in Japan. I just got 2 days ago, but I haven't read yet. So this is the only publication about copier we have. Some of them try to apply for cosmetic, but still not applicable for industry. It's not much information about copier we have. So still huge opportunity for us to work. Thank you.
So I agree with Professor Sisunandar about the spatial temporal accumulation of different metabolites. Also included the health promoting metabolites. So as he had mentioned that the data may be from at the end of last century, right? So at that time, there's no word term of metabolomics. The metabolomic actually started at the end of last century to maybe about 1999 or 2000, something like that. So it's not too long. So we have this method. I think we can apply to the samples that you have, right? So this is the related to a question about for that, and also for the preparation of the samples. There are two ways that we can do that. So the first one is if we have the fresh sample, normally we put it into liquid nitrogen, and we grind it in powder, and we then weigh them, and we then analyze with normally LC-MS or GC-MS, right? That's for the fresh sample. But if you want to keep it for longer and easier for transport, what we can do, we normally freeze dry the samples, right? So you freeze dry the samples, and then you keep the samples maybe at room temperature, but even better at -20 or -80. It can keep for years. And then if you want to, you can grind it into powder, and then at room temperature this time, and you can measure the metabolites or do metabolomics. So it's not that difficult, right? So is that okay? Yeah? Thank you.
We still have time until 11:30, so I would like to encourage students to ask questions. Yeah.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Misan Amalia Hasanah dari Prodi Agroteknologi Angkatan 21. Izinkan saya menggunakan bahasa Indonesia. Di sini kan kelapa merupakan tanaman yang penyerbukannya dibantu oleh serangga, seperti lebah atau mungkin angin gitu. Yang mau saya tanyakan, bagaimana cara menjaga supaya budidaya kelapa kopior tetap murni mengingat penyerbukan kelapa dibantu oleh serangga, angin, yang nantinya membuat kelapa kopior diserbuki oleh tanaman kelapa lain yang ada di sekitar perkebunan tersebut. Terima kasih.
Pertanyaan untuk Prof. Sisunandar.
Ya.
Okay. It's a question. Sisunandar.
Should I use Indonesian or English? Bahasa Indonesia apa bahasa Inggris, Pak? Oh, bahasa Indonesia. Yeah, you said. Okay. Coconut has been divided to two things. Dibagi dua, dwarf and tall, jadi genjah dan dalam. Tall untuk yang kelapa dalam, yang tinggi, yang besar. They usually cross pollination. Biasanya nyerbuk silang. 95%, 90% of them cross pollination. Why? Because when the female flower, ketika bunga betina open, terbuka, all the male flower is gone already. So it is why it should be cross pollination, harus serbuk silang dari pohon lain, from other tree or from the next flower. It is fertile. Case for dwarf is different. Jadi kalau pada kelapa genjah berbeda. When the female flower open, there are still some male flower on the same inflorescence. Jadi ketika bunga betinanya membuka, bunga jantannya masih ada untuk satu bunga itu. Jadi 95% of dwarf kopior, 95% dari kelapa kopior yang dwarf itu nyerbuk sendiri, self pollination. So, even kopior or other coconut is using wind or insect for their pollination process. Jadi meskipun pakai angin atau insect untuk proses polinasi, but because the female flower ready and there are also male flower ready, it's just close, very close, sangat dekat, maka Angin pun juga akan nyerpuk dia sendiri. But not with the tall. Tapi bukan yang kelapa dalam. For the tall usually should be planting in a wider area. So, jadi kalau mau nanam kelapa kopior yang dalam, harus areanya yang agak luas. Not only one. Jadi kalau agak luas, even they do cross pollination, they'll be crossed by other kopior, not by other normal coconut. Jadi kalau karena luas, tidak hanya diserpuk sendiri, serpuk oleh yang serpuk sendiri, tapi sesama kopior. Kalau sesama kopior nggak ada masalah. I have my plantation just next to the normal coconut. Yes, the percentage of the normal increase compared to the area that isolated area. Jadi kebun saya itu bersebelahan dengan kelapa normal, tetapi memang persentase kopiornya sedikit turun dibandingkan yang isolated, yang jauh dari kelapa normal. Turun, the decrease is about 10%. Jadi turunnya sekitar 10%-an. But we still produce kopior coconut. Even the next coconut is normal. So it's not really a big problem if we planting normal and copior just next of them, then it will decrease all copior to become 0%. No. Jadi naturally ya begitu. It's happen naturally. So we can't prevent them. But when we do cross pollination, when we do hybrid program, ketika kita mengerjakan program penyerbukan untuk produksi hybrid itu, for hybrid producing coconut, then we have to prevent them. We put protecting bag, so we use bagging. Before the female open, we cut them all the male flower, put bagging, and then we do hand pollination, put bagging again, and then after 10 days we open the bag. Jadi agar tidak terjadi penyerbukan yang tidak terkontrol, kita taruh plastik, kita taruh bag, Mbak. Jadi kita tutup. Sebelum bunga betinanya pecah, kita buang jantannya, terus kita tutup, kemudian kita serbuk pakai tangan, kemudian kita tutup lagi. Setelah itu, tunggu sekitar 10 hari dibuka tutupnya. Itu ada sekitar 5% lolos. Jadi, even we already do baking, we still have 5% that fail for cross pollination, for hand pollination. So, very interesting. The best one is try it.
Thank you. And maybe one more question from a student or colleagues, maybe who are interested to continuing their study to Hainan University. We have Professor Roger here, ready to answer your question. So please, I would like to encourage student or college to ask.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Hesti.
Izinkan saya memakai bahasa Indonesia, mungkin dari moderator bisa membantu untuk mentranslasikan. Pertanyaan saya untuk Mr. Roger, kita tahu bahwa kelapa itu sering ditanam di dataran rendah. Namun ketika ingin ditanam di dataran tinggi, seperti contohnya kayak Dieng itu kan dataran tinggi. Apakah mungkin di Cina udah dari penelitian Profesor Roger sendiri telah menciptakan suatu varietas kelapa yang bisa ditanam di daerah tinggi? Ya, terima kasih.
Baiknya saya yang jawab, Mbak. I will ask the question. The question is there any coconut in China? Yes. There are a lot of coconut. Kelapa itu, Mbak, tumbuh 23 sampai 25 derajat lintang utara sampai 23-25 derajat lintang selatan. Bagian selatannya China itu masih di antara daerah-daerah itu. Kalau lebih dari itu nggak ada kelapa. Jadi kalau di Australia itu bagian utara banyak kelapa. Di Cina bagian selatan banyak kelapa. Jadi it's not about special variety, bukan karena varietas khusus. Ya kelapa itu hidup di daerah situ. They have tall, they have dwarf. Mereka punya kelapa dalam dan juga mereka punya kelapa genjang. Sama seperti kelapa kita. Nah, kelapa dataran tinggi itu mayoritas kelapa dalam. Kenapa? Karena kalau kelapa genjang nanti tumbuhnya agak berbeda dan produksinya tidak akan banyak. Itu makanya yang dataran tinggi itu banyak kelapa dalamnya, akarnya lebih besar, dan dataran tinggi umumnya tidak subur. Akarnya lebih besar, batangnya lebih besar, maka dia lebih kuat daripada dataran rendah yang genjang, daripada kelapa genjang. Akarnya kecil, makanya tidak banyak tumbuh di dataran tinggi. Jadi kalau punya maksud dataran tinggi berapa, Pak? Di atas 600. Kalau masih 400, 500 masih oke kok. 450, 500 itu batu raden, itu masih oke. Ya, ke atas lagi, nah kalau sudah di Dieng ya enggak ada lah, karena Dieng itu sudah 1000. Jadi bukan special variety, Mbak, untuk itu. Ya kelapa ada di sana banyak. Hainan is the most province that produce coconut. Jadi ada di Hainan itu kelapa banyak, bagus, Mbak. Jadi gitu. Jadi bukan riset gimana. Ada lagi?
Izin Pak Ali, kalau tapi apakah ada kemungkinan ketika ingin memperbanyak produksinya gitu, meningkatkan produksi dari ketika yang kelapa yang tal itu bisa diproduksi lebih maksimal di daerah tinggi tersebut, Pak?
Daerah tinggi, the highland itu, mostly less sun. Sunlight is less, and mostly the fertile of soil is less compared to the lowland. The third one is the water is less compared to the lowland as well. So it is why highland produce coconut less compared to the lowland, because coconut need water, coconut need fertilizer for their normal growth. Jadi dataran tinggi umumnya sedikit produksi kelapanya. Kenapa? Cahaya mataharinya kurang. Jadi kalau agak siang sore sedikit sudah mulai agak gelap-gelap. Cahaya matahari kurang, fotosintesisnya turun. Fotosintesisnya turun, produksi buahnya pasti turun. Itu cahaya matahari. Yang kedua air. Dataran tinggi pasti airnya kurang dibandingkan dataran rendah. Karena dataran tinggi itu baru ada air mungkin kedalaman sekitar di atas 50 meter. Sementara di dataran rendah 1 meter, 2 meter sudah ada air. Dan akar kelapa itu paling panjang 3-4 meter, Mbak. Jadi nggak sampai di air. Terus kesuburan tanah di dataran tinggi umumnya lebih rendah daripada dataran rendah. Makanya kelapa dataran tinggi lebih sedikit dibandingkan produksinya dibandingkan kelapa yang ada di dataran rendah. Itu yang membuat mayoritas kelapa dataran tinggi juga kelapa dalam, bukan kelapa ganja. Itu Mbak.
Cukup ya, terima kasih Pak.
Ya, terima kasih Mbak.
One more question. We were dominated by female questioner. Oh yeah, good timing. Okay.
Assalamualaikum warahmatullahi wabarakatuh. And good morning. My name is Ananda Rizky Martopani. I'm from the Agrotechnology, from the Agriculture and Fisheries Faculty. And this question is aimed to Professor Sisunandar and also Professor Jie Luo. If you could answer, please answer, guys. Maybe this is related between your material that you've presented to us before this. So my question is, from what we've discovered that coconut could be used in kopior, and it is discovered that kopior coconut has a lot of galactomannan that could be used in cosmetic. My question is there any possibilities that we could create through CRISPR-Cas9, or maybe we haven't discovered the secondary metabolism from the coconut that it could be used for something that we could not know? Because for me, the idea of kopior coconut is already mind-blowing. Like, I live in the small island, and there, if you find coconut, there wouldn't be kopior coconut. And here in Java, I discovered that turns out coconut could be the kopior coconut. So that's my question. Is it any possibilities that it could be used to another purpose that we haven't discovered, or yeah, anything else like that yang which mind-blowing? Because what I've been discovered that what the world and what China has makes for these recent years is for me so mind-blowing. Like, I could never think of think something like that, like it is so mind-blowing. So is it possible? Thank you so much.
So the question is it possible to cultivate kopior in other island or other province? Is it that I'm right?
Sorry, no. I mean, is it possible that kopior being used to discover another secondary metabolism in it? Because we've already discovered that it has a lot of galactomannan. I mean, could we discover another chemical maybe that could be used to another thing like that through CRISPR-Cas9 or maybe anything like that, to that in the future we could use it to something.
Okay. Is it possible to produce to identify another metabolite rather than galactomannan or other we can have? Yes, of course. There are plenty specialized metabolite. We usually use secondary metabolite. There are plenty specialized metabolite in copper as well. Possible. Because we haven't done yet. We haven't finished yet. So if you are interested, then this will be very, very, very useful of your work for other people to work with. It's very possible. We found galactomannan, we got Buvinar team got tocopherol. So there are still plenty metabolite that we can working with.
Yeah, just a brief. So if you want to, if you're interested in a different secondary or special metabolite, what we can do that in addition to copia, we can analyze different varieties to get to the diversity, maybe something not in copia, but in other varieties or accessions, which is maybe health promoting, or you are interested, you can introduce them into copia, all right? That's the traditional way. You can, by crossing, something like that, you can do that. But we are also developing the method for transformation. And then you can use the CRISPR-Cas9 as the strategies, as you mentioned, but we need to set up the method for the genetic transformation first. So Dr. Mu is doing that, and we would like to get help from Professor Sisunandar to set up this tissue culture and the transformation protocol, right? To make it possible for transformation, to do transgenics in copia or in coconuts, right? Is that answer your question?
All right,
Yeah.
that is very answering my question, and for me it is very interesting. Turns out, from the coconut alone, we could discover a lot of things. Thank you so much, Professor Sisunandar and Professor Jie Luo. Thank you so much.
Yeah, thank you for your question.
Thank you. Thank you very much for the participant for all of the question. Again, once again, please give an applause for the our speaker this morning. Thank you very much for Professor Roger and also Professor Sisunandar for sharing your knowledge to us today this morning. And then with that, I think I would like to give back the floor to the MC. There will be a souvenir handling for the speaker. Is that correct? Yeah.
I don't know what will happen with us.
Thank you for the moderator. And to pay tribute to our speakers today, we have prepared special souvenirs for Prof. Roger and also Prof. Abraham Mo. And maybe you can give it to them. And also a certificate for our speaker, Prof. Nandar. And also the certificate for Ibu Ika. Ibu Ika? Thank you to our speaker. You may come back to have a seat. Bu Ika? Ibu Ika, you may come back to the stage for certificate giving. Okay, ladies and gentlemen, we have now come to the end of the seminar. Thank you for coming to this international seminar. We would like to give a big thanks for Professor Jie Luo and Professor Nandar as our speaker today. To close today's seminar, let's recite Alhamdulillah together. Alhamdulillahirabbil alamin. Thank you and see you next time.